I will try to be brief. I know I'm standing between us and dinner. And I'll also try to be interactive. This has been a long day, and we've heard a lot of different things. And what I'm about to talk about, I'm not going to lie, is not exactly the most straightforward. It's about an European project called Cure 2020. Standing for uveal melanoma, finding a cure or hope for a cure until 2020. It's a European Union project. It's working? Okay. So it's an Horizon 2020 project. This Horizon 2020 has been a large initiative from the European Commission. The UK was still part of the EU back then, and there are 12 partners in Europe working for these efforts. The 12 partners received a €6 million grant, and this grant. Okay, so it's this way. This grant has several ambitious, important objectives. Okay, there you go. I was thinking about whether I would change this METS into uveal melanoma metastasis, but I thought that you see that these things get called different names, so I decided to leave it. When we talk about it's uveal melanoma, and many times, and I'm sure you've heard this in clinical settings, people talk about METS instead of metastasis. And so our objective is to better characterize this metastasis, and I will go a little quick, and then I'll come back to these themes. Then evaluate either single or combination of drugs, and we talked about drug combinations here today before, to identify new therapies for metastatic uveal melanoma, which is one side of this disease that still does not have adequate treatment. We want to further develop, and this is where we will be talking a little bit about what we call basic and preclinical research. We want to develop relevant in vitro and in vivo models to understand the mechanism. So one of the reasons, or the main reason why we do not have fully successful treatments, is that we don't know much about this disease. It is a rare disease, and we need to understand it in order to really tackle it. So we want to characterize it and to use research models to do that. We want to be able to see what changes in the disease that leads to the metastasis process and what makes metastasis aggressive, so we can identify new targets, new drug targets and pathways that we can really use in order to find new treatments. And finally, and you are a very big part of this step, we want widespread dissemination and maximal exploitation of the results. We don't want them to be hanging on a shelf. We want to really get them quick into a clinical setting, and we want patients to be involved even before the results are there. And I have to say, just as an aside here, this project really has been humbling. I mean, just hearing your story right now, and it's been a huge a great experience to have the involvement of patients at all stages of this process. And I have to say, we're 12 partners all across Europe. Some of them are here. And if there's one thing that really unifies very different personalities, very different countries, very different cultures, but one thing that does unify all of these partners, scientists and clinicians alike, is the wish to find new solutions for this disease. Okay. So the main approach of the consortium is that we will depart from patient-delivered tissue and then do the research. So many times people are looking for pathways for causes of disease that are the causes of many diseases, so that they can then find something that will then be of use in many different conditions. So because this is a rare disease, right, one of the limiting factors to finding out more has been the lack of availability of the disease, of the tumor tissue itself. And so the project really puts the patient at the center. And again, for the sake of speed, I will explore this in the next slide. This is the worst slide of the whole presentation, but I think I needed to have this. So there's no board, but we all understand what cancer is. We all understand what tumors are. They're these cells that are a little bit like, initially like normal cells in different areas of the body. In this case, we're talking about cells in the uvea, which is this part of the eye that encompasses the ciliary body, the choroid, and the iris. And these cells are there, and something happens that makes these cells go astray and start dividing too much. Okay? We think of cells as spheres, but cells really are these living spheres that are hearing the world around them, and they're communicating with the world around them, and that regulate whether they sit, whether they change shape. And so cells have, so if you think about this, I've just cut through a cell, and this is the circle, and cells have on their surface all these receptors that allow them to hear the environment around them. And normally cells know that they differentiate, they become part of the uvea, and they have a function, and they don't do anything else. Now, what can happen, what happens in cancer, is that you have this tumor growth and proliferation. Oops. And so what happens is that the mechanisms that cells use to just be quiet somehow go astray. And what this illustrates, and it sounds very complex here, is that something in the pathway from these receptors that are outside the cell into the things that the cell coordinates biochemically and in its nucleus to turn genes on and off and decide to proliferate goes astray in cancer. And different cancers can have different sorts of pathways disrupted. For instance, we talked about how uveal melanoma, ocular melanoma, is different, or the uveal kind of ocular melanoma is so different from skin melanoma. And that's because the pathways that lead to this proliferation are different. And so pathways are what? They're all these proteins that are lying there and that communicate with each other and change shape. And they can be phosphorylated or not, and change shape accordingly, and they can be activated or deactivated. So a lot of the things we're looking at when we're looking at medications are things that are acting in these pathways, that are activating or inhibiting pathways like this, either at the receptor level, at the level of the thing that is outside of the cell and hearing the world, or internally in the cell. And so a lot of the research that goes into looking at how different this uveal melanoma is, how different these metastases are, will also go into looking at these pathways. And of course, because these pathways are not the most common in the most common cancers, they haven't been very researched. And so it's been really important in order to be able to have enough results to initiate promising clinical trials, to set up a system across Europe with several partners that initiates where? It initiates in the metastatic tissue. Not just metastatic tissue, but in the biobanking among several reference centers of these disease samples, primary and metastatic disease samples, and then the sharing of these samples in order to do research, to find new results that allow us to test new medication. And so these models we were talking about initially, they are what? They are living models. They can be what? They can be cells in a dish. So you can culture cells from a biopsy and develop a cell line that is similar. It's not exactly the same because it's not in an organism, but that is similar to a tumor. And it grows in a dish. We need to feed it, but it grows in a dish. And in many ways, it mimics what's happening in a tumor. Not perfectly because it's in a dish. You can then look at this and study these in organisms, and I will show you how. In classic ways and in new ways, and we'll explore them, and then try to characterize, now that you have all this metastatic tissue from all these people, and there's some people here, Sarah Copeland will be talking tomorrow from the University of Liverpool, is going to be talking about these biopsies. And then these are the biopsies that Sarah, or some of the biopsies Sarah will be talking about. And you can, now that you have the tissue, you can characterize it, get to know better, and finally you test. You don't just test single drugs, and you saw my question earlier about the triple therapy. You test, you get promising results, and then you already look at combinations so that you get the most promising clinical trial coming out from these results. So it's kind of complex. It has all these partners. Now, how are these novel approaches and research models done? So you can take cells from a tumor, and this looks a little bit of sci-fi, but I think it's interesting to know how these things, and you can inject them in a mouse, and these cells will actually grow and attack the mouse tissues. Now, you can imagine that the tumor growing in an organism is much more alike than the tumor just growing in a dish. So this allows us to look at it with a different kind of rigor and a different kind of exploration. You can also do this in a fish. And I've inserted a couple of slides because several of you asked a lot of questions about this in a previous meeting. So you can do this in a zebrafish. And what you see here is a zebrafish with human tumor cells in it. The way you do this is these fish, I'm looking at the dish from above. These are larvae. And this is a fish larva with tumor cells. And you can analyze them. So in red, the tumor cells, and in green, their vessels and some autofluorescence. And now this is not uveal melanoma, but this is an example on how human tumor cells injected in a fish can actually then invade distant sites for these kind of studies. And then you have to look at whether these actually are still alike the human tumor. And in many cases, these have been proven to be so. So you need to test these things and then validate them along the way to make sure that we're still looking at something that is like the human tumor. And this is how you do the first stages of research. It takes a lot of time, a lot of people, a lot of animals, and that's why a concerted effort really is the way to go. And then you can imagine, and this is also not for UM in itself, you can get many of these inserted, and then you can test for different drugs in a small animal. So these are some of the novel approaches that you can use to quite quickly test good candidates for therapies. Now, this all looks great. There's all these people. So there are expected results, and we're expected to deliver very clear results, and we've already delivered some of these, okay? So we divide the work, and you saw all of you, if you allow me, have in your tables, because this kind of gets super complex, both a save the date, and there will be a meeting in Liverpool, and this schematic that I had in an earlier slide, but I can't have on all the time, where what we're looking at is the flow, and it does go back and forth because you have to test things, the flow of research within the consortium. And we divide it in work packages. So you see WP1 is work package 1, WP2 is work package 2, and they interact with each other. So WP1, you have to have a networked European virtual registry of uveal melanoma biosamples with harmonized SOPs for samples collection biobanking. SOPs are standard operating procedures. It means that the British have to agree with the Dutch, that have to agree with the Polish, that have to agree with the French. Not always easy, but if everyone is determined and sitting around a table, you do get it going. Because if we're all treating the samples in a different way, then how can we compare them, right? And so this is important. And then we have this preclinical evaluation of biology-driven therapeutical approaches, another big title. What that means is that you need to validate these in these mouse and fish and cell line models in order to go forward. Then you develop this next generation preclinical models, and I just showed you, for example, these fish. They can be this PDX, which is patient-derived xenografts. They can be CDX, which means circulating tumor cell xenografts. They're tumor cells that you take from the blood. And then they can also be GEM models. For example, they're genetically engineered mice that we engineer them with the mutations that we know occur in UM. And if you do that, then we observe the kinds of changes that occurs in the mouse if we change the genes that we know change when disease happens in human. And these are important. Sorry, I have a little trouble. You characterize the metastasis, and we want to evolve to develop a chip where we have developed a chip that looks, remember those proteins? And those that we saw in the first slide, that looks at those proteins, for example, and their state, whether they're phosphorylated or not. And so that we can kind of correlate that with disease, among other factors. And we need more detail on the pathophysiology of uveal melanoma. And Sarah Copeland. I am not myself involved in the research. I'm involved in work package 5, which is the dissemination and implementation through clinical trials initiation. So initially we're working with MPNE for the formation of the European patient network. And towards the end of the project, we want to be able to have enough results and enough networking and enough expertise that we can actually draw up European guidelines for uveal melanoma patient management. And you should see these guidelines across the whole continent, and if you're including much of Eastern Europe, are really not there or not being followed, and it's really important to kind of push them, not just write them, but then help them to be executed. And in the UK, you have been very lucky with the state of affairs so far. It's not enough. It's not maybe what we want, but it's certainly much better than many other places in Europe. And finally, we want to have at least one clinical trial initiated. So this work package 5 that works with patients, I'm going to go quick here, aims to develop this patient community. Eventually, as we have results, we want to interact more with companies in order to initiate clinical trials and to bring them to patients quick. There are 12 members. 4 of them are referral centers that see patients. Institut Curie, which is the major referral center in Europe, in France. The University of Liverpool, here in the UK. Leiden University Medical Center, and Jagiellonian University in Krakow, Poland. These are important centers that see a lot of patients in uveal melanoma that have a lot of expertise in clinical care and the pathology of the disease. Then 3 research institutes, among them the Cancer Research UK Manchester Institute, Leiden University, and Trento in Italy. 2 biotech company, Spep Therapy, that develops these peptides that attempt to inhibit parts of those pathways when they go bad and start driving proliferation, and PAMgene, which is developing this chip looking at the state of precisely those. Then the Champalimaud Foundation, which is where I come from, and we are working with the communication, dissemination, and really trying to get the project to you, and the communication within the consortium, and the meetings that the consortium also needs. The Melanoma Patient Network Europe, Bettina Rill and Ian Galloway, now Andrew Evans, Dick and many others have really joined forces, but this was actually even difficult to find initially. We wanted to have patients in the consortium, but we were looking, and Ocular UK has really played a very important, where's Joe? There, a very important role in being present at all of these activities, and finally a company that works with this. So this is where we are, but we aim to serve the community at large. The stakeholders are, of course, the uveal melanoma patients themselves and their families, the oncologists, surgeons, ophthalmologists in charge of the treatment and management of these patients in a clinical setting that may not be as well informed, the global scientific community, and finally also industry. I have to. And so we have this set of communication tools, and I would really, and I will go through this, I would love you to look at the tools. You have our website, you have this, and we really want your input. The website has dedicated areas, so you can explore different aspects of the project, and for us it's always very important to have feedback. We've developed these consent cards that some of you might already have seen, where patients that are actually new patients whose metastatic or primary tissue is participating in the consortium are given these consent cards, so they know more about the project. And also, if they change physician, they go elsewhere, they can bring and say, like, I actually have my tissue within this network, and the person can contact, in this case, Sarah Copeland in Liverpool, and so on. So this is really important. These meetings are really important, and your feedback is really important. You will probably see a little bit more of this with Bettina Rill tomorrow. I have to say, and Ian, it's really been a privilege working with you. When the consortium started, Ian was actually still not involved, but he got involved very, very early. And there were many fewer patients in many fewer countries, and it's been really expanding. And this has really been important, and we hope will prove really vital for the next stages of the project. Now, clinical trials, how are we going to get there? We need to look at these results. There's a scientific advisory board that meets yearly and that looks at these results, having in view which results are we going to select to take to clinical trials, to take to the next stage that eventually is going to feed into clinical trials. And the translation of this preclinical data to the clinic is really important. So you have a part in this project. I thought I had more time, so I would do a little bit more of a discussion, but Guilherme is here. Guilherme is an intern in ophthalmology and is working with the project within a fellowship. So your input for the communication tools, for how this network is taking its role within the project, and then now with what we hope will be a useful patient brochure that we have samples of, and we would love your input on, and we want to distribute widely, precisely so people can take it home, use it to discuss it with the physician, use it to discuss with their families, and understand the disease and both the primary and in the metastatic setting in a way that is clear and provides good, useful, up-to-date information. And finally, and we just heard from Thomas, I think he had to leave, right? In participating and providing input in the design and initiation of the clinical trials themselves. So the way they are taken into the clinic is something that is of benefit for all, and that really takes results to the next step as quick and as effectively as possible. So the project, in order for the project to work well, there need to be all these meetings, and every meeting. So we have this general assembly, and every meeting there is a patient meeting associated with it, where different aspects of uveal melanoma are communicated, and also some of the results of the project are discussed. So what you have here is the save the date for the next meeting. This was the kickoff meeting. This is the first General Assembly. And the next one is going to be on the 30th of November in Liverpool, and we hope to see many of you there. The Scientific Advisory Board are composed mainly of people outside the project and from different parts of the world. And these are the people that are yearly scrutinizing our results and helping us move forward. These conferences that bring everyone together, just patient conferences themselves, have really proven very, very important and useful. MPNE participates in them. The last MPNE Ocular Rare conferences in 2017 in Leiden. We've now had a second advisory board meeting. Bettina is sitting there, always looking fairly incredulous. Which is great input. Andrew is here. Ian's probably sitting somewhere over there. And what we also have tried to do, not necessarily as a consortium, but as consortium members, is bring the community together. At the foundation, we just had a large international meeting. It was the first time that ARVO, which is the Association for Research in Vision and Ophthalmology, that brings together almost 15,000 people a year, had a meeting that included patients. And we just received the feedback forms last week, and it brought together both oncologists and ocular oncologists. So general oncologists, ocular oncologists, ophthalmologists, all together discussing the most recent advances in the field. And what the feedback forms say, it was great to get patient input. It's really spectacular how important that was, and how that brought people together, because the guidelines are different from country to country and from the EU. So here's the next meeting. It will be our second General Assembly. We will have 4. So this will be the second part of the project being discussed, moving already into the third and fourth part. So this will mark almost halfway through the project to the clinical results. And I want just to bring here some important data. This is not necessarily part of the Cure initial. This comes from earlier on, but these have been important results, and we've talked about them here. You can see them and explore them on our website. What these results that came out in Cancer Cell show really is the stratification, the risk stratification of the tumor, and the primary tumor, and what that means for the risk of developing metastasis. And you can explore this in the website. And I think that it has both a video and lots of different levels of discussion to look at this. We've talked about monosomy 3, so the loss of chromosome 3 as one of the drivers to higher risk of metastasization. And that's here. Then there's also chromosome 8. There's this BAP1 mutation, several of the players in those signaling pathways. As we work comparing these data sets of metastasis, we have found, and in this case Institut Curie and the team there, found that a small percentage of patients, but still a percentage of patients, has not the very low mutation rate that was discussed here earlier, but a higher mutation rate. So they saw that this lady, 79, that presented, so came into the clinic with metastasis in several organs, was responding to immunotherapy. And she responded really well. And you can see and compare here that where there are metastases after treatment, there are none across the 4 organ types. And so they found that there was this mutation, in particular MBD04, and the mutation rate in itself that was related to it. But even if it's a small percentage, you know, within 100 people, there's 2 that can be helped effectively. And we know which ones can be helped with these therapies versus the ones that might need some more. And then there's also a discussion of this review from Liverpool, which discusses recent breakthroughs in metastatic uveal melanoma, and discusses them at large, and not just within the research of the consortium that I invite you to read. Now, Guilherme is going to be here, and I'm not going to spend time seeing this, but we had a vote in one of these meetings on how we would have this patient brochure look like, and everyone wanted something small that you could fold in 6 and you could take home in your pocket, but everyone also wanted something that would be broad and talk about everything. And so we tried our best to do something good. This is not how it's going to look like, so the design will be better, but you will see some black and white sheets where we're discussing this brochure. And we really want your input on whether you find it helpful or not. We have something, we kept the eyelids so it really looks like the eye, looking at the eye anatomy so that you can have a conversation with a physician, but also examples of uveal melanoma, and then one example that will have a different color of conjunctival melanoma, which is a wholly different disease. And then take-home messages, you know, that you can read one at a time, and then again, this is folded in 6, so it's also easy to explore and to digest. I'm referring to your talk before. And then on the other side, we will be talking about liver metastasis and what can happen. So please, if you have a chance, can you stand, Guilherme, so everyone can attack you at one point? So Guilherme will be here, and I will as well. And we have several black and white copies of this. And we would love you all to explore and give us lots of feedback, write on it and just give it back to us, because we really want to incorporate the most possible the consensus on how to make this useful, and then to distribute it as widely as possible. November 30th, please keep this on your calendars, and you can come to Liverpool. There will be catering, there will be travel grants available, and we really hope to be able to to see many of you and have your input in order to discuss it later. So you can follow us on social media, both Facebook, Twitter, and YouTube. We try to have a lot of videos explaining different aspects and discussing different aspects of the disease. And you can get to all of this from this website that you also have on this page on your tables. And so, if you have any questions, I'm here for them. Thank you.
consider doing a brochure specifically designed for ophthalmologists? Because there are many patients who are complaining about superficial exams, their tumor being detected late.
Yes, so what we try to do, and you will see that in the text, so we wanted to do a brochure that would be centered on the patient, but that would also have this information that many times ophthalmologists do not have. So the reason you have that picture that is so anatomical of the eye, and you will have the uvea here even more evident, is precisely so that it appeals to ophthalmologists, and ophthalmologists also get some of this information. So you have possible signs and symptoms, you have methods of detection, you have treatment choices, and you have the need to refer the patients to specialist centers. All this is information that ophthalmologists also get. But we want this to be a patient brochure, so the patient can take it to the ophthalmologist, can take it to the family, take it to a second opinion, and it's something that can be navigated in that way.
Just to clarify, I think to answer your question, the main goal of the brochure is to inform patients and to give them pointers on which questions they should ask if the ophthalmologist or the oncologist doesn't initiate the conversation, him or herself. It's an attempt to give the same standard of care across different referral centers.
In different settings overall.
Presumably, as you set this up with a pretty ambitious goal,
yes,
one of the things I guess you would have done was a review of the literature globally and, as it were, non-European Union academics and clinicians.
Of course.
Because what I'm interested in trying to help kind of bring about is almost a sort of map, if you can imagine it, of what we know about the basic science of the disease and what we don't, and try and get that into one place. With Joe is sort of involved, well, very much leading on this, but try and get that into one place so that we can then essentially from a patient start to influence from a patient perspective where is more research needed, where are the unknown questions? Because one of the things I've found really surprising as I've got more involved in sort of understanding research is it's all a bit competitive and a bit random and a bit, and I know that sounds disrespectful, it's not meant to be, but it just feels from a patient's perspective as if a little more collaboration, which I think you guys have been exemplars of, but is unusual, I'd say, and a little more sharing of what's already known would make for more efficient research and then ultimately get to quicker trials, because the basic problem is not enough is known. You said it right up front, not enough is known. So I suppose it's kind of, would you be in a position to share with a patient group essentially your mapping of the landscape that you must have done as part of setting up this whole project in the first place?
Absolutely. So our results are reviewed yearly for each one of those packages, are reviewed yearly on a scientific advisory board whose members are experts from all over the world, from across the ocean included, and where patients sit and are a full part of this committee. So we do that regularly. And we also try to organize meetings, both within the consortium and then the individual partners themselves, that bring people together. But you are right, the way science is organized appears to be many times a little random and not always focused on communication. And we have to understand that the incentives for the different professions that make science go on are different. And understanding that kind of create the structures that allow for sharing. So scientists advance in their careers with the published results. So they don't have much incentive to be sharing unpublished results that then someone is going to be using. So, and this is understandable, but what we want is for things to move fast. And so there needs to be this trade-off. And so we certainly do a lot of sharing within the consortium, and we get a lot of input and discussion outside the consortium. But you're right, that's really important. You don't want to be repeating. The worst thing is putting a lot of money into doing something that has already been done and shown not to work. And many times people don't publish negative results, right? They tried something, it didn't work, they didn't publish, nobody found out. Someone else is going to put a lot of money into doing that, and that's definitely